Welcome to this Unit 4. And let me just give a 1 minute like a summary of what we have done previously. We studied the gene prediction in Unit 1. And then in Unit 2, we used the software programs to predict the genes in prokaryotes. And in Unit 3, we tried that for eukaryotes. And then, and do you guys remember that we did a classroom exercise together to get the sequence of a gene using which one? NCBI, NIH one, right? So in genomics, in the United States, NCBI NIH is a big like an institute that offers a lot of database like that, but that is not the only one. You probably heard the University of California, Santa Cruz. They offer the UCSC, the genome browser. So you will learn about that in the future. But today, you will learn about mostly on the European database. So something comparable to NIH in US. So everybody, is there anyone who knows that something comparable to the NIH in Europe? Have you ever heard the EMBL? Nobody heard it?
Yes.
Yeah. So let me just introduce that. European Molecular Biology Laboratory. They have bioinformatics, what is that? The subset or institute, which is the European Bioinformatics Institute. So, in US, NIH is like a 28 institutes get together, and NCBI is the subset of that one institute that offers this database. In Europe, the most famous one is the EBI. Here, the European Bioinformatics Institute, EBI, from the European Molecular Biology Laboratory, EMBL. So we will basically, you can expect something similar being offered in NIH as well as the European like counterparts. So sometimes the NIH data are more accessible and easier to understand, and you can quickly get the result. But sometimes the opposite is true. The European one, the Ensembl database, is sometimes a lot easier, and then provide the right information. So at this time, it actually depends on. So as like a genomic researcher, I wanted you to know all these three major sources, how to access and how to retrieve the biomedical data, especially bioinformatics data, from not only from NCBI NIH, but also from the Ensembl database from the European institute, as well as finally we will touch on UCSC probably in the future. So do you have any question on the this big picture? No? Right? Now, I would like to do this in person, like the lecture, more like the hands-on driven or the question driven, so that you can actually actively participate. And then I would like to see what kind of mistake you guys are making, and then we can discuss it and we can fix it, and then or yes, like that. So I would like to apply some more like a hands-on, not just a boring lecture. So, and okay, let's get started. How many coding genes in human chromosome 11? You're probably tempted to use a ChatGPT. but I want to have updated information. ChatGPT 2021 is the latest information, and that number is outdated. So I want you to have provide the most updated coding gene numbers in human chromosome 11 with the reference. According to this reference, the number of coding genes are okay. But while you are thinking, let's give you some thought. How many genes do we have in human? 20,000, right? So it's always a good idea to have some general understanding. So we have 20,000 genes. How many chromosomes do we have? 20 to 23 or 22 autosomes and then X and Y. So basically 23, right? 23 pairs. So if you divide by 23, it will give some rough, like an estimate. And if you check the number of genes in chromosome 1 versus number of genes in chromosome 22, do you know the difference between that? Chromosome 1 is pretty long. Chromosome 22 is the shortest, right? It's ordered by the length. So number of genes, you will get the more genes in the chromosome 1. But chromosome 11 is around in the middle, right? So let me, I estimate, if I just estimate by 20, so around the 1,000 genes, coding genes about that order. Probably 800 to 1,400, that's my guess. Because it's not the exact calculation, but I still want you to have the way how to estimate before actually Google or search. So I need to give the couple of minutes, and then let's why don't you actually try to find the how many coding genes in human chromosome 11, and if you found that, I recommend you to share your screen and then tell me how you find it. And if someone else finds it in a different way, also share it and we can discuss. Yeah. The only thing that doesn't count is a ChatGPT. And I hope that you can find it using the Ensembl. And let me see. You found it? Yeah. How many of you find it? Okay. What is your like a finding? How many?
1, 2, 1.
1, 2. Let's do this. Okay. Let's try this. Let's have it. How many?
1, 3, 1.
1, 3.
1,
1, 7. Is there anyone who got the different number? All right. Is there? Let me stop share my screen, and can you share your screen? I'm very yeah. I'm interested in how you did it. Yeah. Could you show me the other steps that you have done from the beginning?
This is a paper, so I just went to the
Oh. Yeah. Okay. That's a good way. Is there anyone who found a different way? Yeah. You basically found the a link to that, right? And then that's okay, right? There's nothing wrong. And I have just one like a thought-provoking question here. Do you think that the link is updated with the database update or it stays same?
It's written in July 2022.
Pardon?
It's written like the current update.
Oh, I see. Yeah. Okay, that's good. Yeah, there's nothing wrong. You can do that. No problem. Anyway, the number is it's slightly over 1,000 genes, 1,300 genes. And yeah, let me actually introduce this. Let me share my screen again. Then did you also find the how many short variants recorded in the chromosome 11? Yeah. How many base pairs? So the question number 2 is question number 1 is 1,317 genes in chromosome 11. Question 2. How many base pairs for chromosome 1? Chromosome 11. What is it?
33,962,300.
Roughly 33 million base pairs.
Yeah.
Okay. Now, that was the first part of the question, and the second part of the question is.
I gave the short variance. The length is 135 million base pairs.
Out of
135.
135 million base pairs. So how many base pairs for chromosome 11? About 135 million base pairs. So do you have to know how many bases do we have in a haplotype? If you add all the bases from chromosome 1 to chromosome 22? 3 billion. So 3 billion. So that's the chromosome haplotype of chromosome 1 to 23 and sex chromosome, roughly. And chromosome 11 is about 135 million out of this. So probably 135 out of 3,000. That's about the ratio, which is roughly 1/20 or something. And then now the number of variants is around the 33 million. So if you divide that by that, it's about
4%.
1/4, roughly speaking.
Yeah.
What does it mean? At this time, we found the variance at every 4 space. That's the frequency. And now I have a second question here. This is the number of the variants we found. This is the total number of chromosome 11. This is the total number of all the chromosomes. Right? Is this number the same between the people, or is it remain the same, or slightly different, person to person, and in the future? It will change slightly, but not so much. How about the number of base pairs in the chromosome 11? It will change slightly, depending on which person you are doing. How about this?
Yeah.
It will change. If it changes, it might be to reduce.
Yeah, these 33 million bases will change a lot. A lot. So the number that you have now, if you compare like 10 years later, this will change a lot. This means very little, and this one is also very little.
So how about if compared between 2 people at the same time?
The average difference between you and me. we are roughly speaking 999 base pairs correct out of 1,000. So that's around the 999 about 1,000 names around between the person to person. So 0.1% of a team. Okay. Now, I want to do this even though you can do the Google search and then happen to have a good luck to find it. But let me introduce that you can actually systematically find it in a database. So let's do that together. So let's do it together. Can you Google Ensembl Ensemble? Do you have that my chat there? So let's do it together. Share screen, desktop. Okay. So Ensembl is the one Ensembl is the one that this one, the European database. So I will go to the www.ensembl.org. Please do it with me together. That's why I'm doing slowly. When you do that, you will see something. How many of you go to the ensembl.org and how many of you are redirected to somewhere else? And have you ever heard of meter site? You didn't hear about the meter site? Yeah, but when you do the bioinformatics, you'd better know these kind of things. The main server, the ensembl.org, is not located in the US. Yeah, it's a European one, right? It's probably located somewhere. And if you actually request the information to that European server and get that, unfortunately, the speed is slow. So is there any like a way that exactly same information reside in the east coast of the US, mirroring the original database, and then we don't go to that original Ensembl site, but we go to the mirror site in the east coast? Then we can get that pretty quickly. So far, so good, right? But sometimes, even though it is supposed to work, everything should work as is, but sometimes some link in the mirror site may not work or like that. So be aware. So if you really redirected it to the mirror site and then some link it's not working, think about, oh, I might want to go to the main site and then try to reach that link. So that's another tip. Okay. Now, the human, and then I select the human. I can go to the location chromosome 11. Oh, sorry. Let me share it again.
Professor, could you please share the slides as well? I think we will get the Google slides in the PPT presentation.
Yeah, I'm going to share it right after. Yeah, because because I put the solution to there. So, yeah, that's why when I ask these questions, the easiest way is go to the next page and look at the answer. So that's why I'm sharing right after the So let me share my screen again. So if you carefully look at this link, do you see that the US East link? Let me just. If you carefully look at this link, do you see that the US East? This actually tells you I'm accessing the mirror site, not the main the European site. Go to the Homo sapiens and location and chromosome 11. That's how you construct the that but when you go there, yeah, here is the link that you found. That's exactly the same thing. So what I did was, I go to the Ensembl and then select the human. And in the search bar, I'm typing CHR11 and go. Then I come to here. Do you have any question to reproducing what I have done? Everybody could come to the Ensembl and check out all the summary statistics. So by doing so, you can actually check the how many base pairs are on the other chromosomes, right? Okay. Now, I would like to use the Ensembl for studying. Previously, I studied the chromosome, right? The chromosome 1, how long is it, and how many genes it contains. But can we narrow down, not just the chromosome, but let's focus on one gene. So today I decided to study the HBB gene. So can you guys search and try to explain to me what is the HBB gene? Which is one of the 20,000 genes. I will give you 1 or 2 minutes. And as usual, I will ask a volunteer to come up and then explain what is this gene, HBB gene is. It's one of the 20,000 genes, but this is one of the very important genes.
It is involved in it codes from proteins involved to transport oxygen from bonds to various chemical entities.
It encodes for what?
For a protein, which is responsible to transport oxygen from the lungs to the various chemical entities.
Yeah, this gene is responsible. It's a protein, like a part of transferring the oxygen. Yeah, that's right. And is there anyone who got the what this HBB stands for?
Hemoglobin, supplementary carbo.
Hemoglobin. Have hemoglobin beta? Subunit. Yeah. And so everybody knows the hemoglobin, right? The hemoglobin has a multiple component in it, and one of the component is the hemoglobin beta, subunit beta, and there should be a hemoglobin subunit alpha, right? So, and so HBB gene is making a protein called the beta globin, and this beta globin is the part of a hemoglobin. So yeah, definitely there is alpha globin too, so which calls the HBA. So there is the HBA as well as HBB, but we are talking about the HBB, which is a part of a hemoglobin. Now, let me share this link. And is there anyone who can try to find the 3D structure of the hemoglobin P, HBB? What does it look like in the 3D structure? A 3D structure of HBB gene product, which is a protein. Can you try this link? Did you get my chat message? This is a model. The software used to generate this model is AlphaFold. And they just gathered all the predicted structure based on the AlphaFold software, right? Shall we go together? Do you see this 3D structure of this HBB gene? And you know the gene is starting with a methionine, ATG, the start codon. And let me click the first amino acid, methionine, is located here. Right? It's mostly the alpha helix. Okay. So now we know that HBB gene is a component comprising the hemoglobin, and the shape looks like this. It's a protein. So do you guys have any question before we move on? Can you ask me again?
So the PDB, the protein data map also has like 3D structures, right?
Yep.
So what is the difference between how it's predicted there and in AlphaFold?
Yeah, this is the database of prediction. It's not the real structure. It is, for example, let's say that, do you remember the Glimmer, the gene prediction software? And after predicting it, you just make a website and let people access the result. So this is exactly like that. Instead of Glimmer, they use the AlphaFold. But PDB is the real. They use the X-ray structure or the NMR to decide the real structure. And why do I show not the PDB but the AlphaFold here? because in reality, when you actually try to get the 3D structure by crystallography, the beginning part and ending parts are usually missing, so you don't get the full picture. So from the real structure, you have to find the best one. So I just wanted to get over that step. So that's why I'm just showing the computational prediction model. But if you want to find out the real one, yeah, you should try the PDB, protein data bank. They predict the structure based on the PDB. Pardon? Yeah, sequence, that's right. Sequence and the PDB, the protein data bank. I just protein PDB structure. So now the protein data bank has this protein data bank is rcsb.org. And yeah, if you look at the HBB gene, yeah, there are multiple, but something is combined with the DNA. So it's not really, so you have to do a lot of search to find the right one. But this is a real. Yeah. Now, the gene is the part of the DNA that code for this protein. And the DNA is transcribed into RNA. Now we are talking about the human, right? And how many kinds of mRNA transcript do we know at this time for HBB gene?
5 mRNA. 6 ones we can.
Yes, that's the according to the Ensembl. And then could you explain the detail of that? So that is the next question. Pardon?
What specific people?
Oh yeah. So is there anyone who wanna volunteer and then explain everything that you see here to your classmate? So what is this red thing? What is that? What is the yellow? What is the blue one? Like that. So I'm asking you to find the HBB gene in Ensembl and try to explain this picture, what you see.
There are 6 transcripts, out of which 5 are coded. The first one has 2 exons, the third one has 3 exons. Second and third both have 3 exons. Fourth has 2 exons.
All right.
They are spliced differently.
And what about the other people? Are you at the same page, or do you need some more time? Shall we check this together? So let's go to the Ensembl. And then search the human. And I want to search the HBB gene. Go. When I searched the HBB gene for human, I got the many hit. Am I interested in the gene level, or am I interested in the each transcript level, the mRNA? At this time, I want to see the big picture, everything. So I want to check out the gene level, because if I click the transcript level, I cannot see any other transcript. I can see only that transcript. So I want to check click the HBB gene. And now this is the HBB gene in the Ensembl. Before I move on, I want to make sure that everybody can come to this page yourself. If you get lost, please let me know. All right. So if you come down, do you see the this kind of picture? HBB genes. Let me magnify slightly. So this is actually the picture that I actually have shown you in this PowerPoint slide. I just magnified it. So why don't I actually go together? Do you see that all this name has started with the HBB? Because that's a gene name. Dash, there should be a 201. HBB 201, HBB 202, HBB 203, HBB 204, 205, and 206. Do you see that the 6 transcripts, right? Let's go to the, let me explain this, the first one, the top one, HBB 204. Let me just check the, HBB 204. And let me ask one question. Is this gene, DNA has a 2 direction, 5 prime to the 3 prime direction, which is what we call the forward direction, or 3 prime to the 5 prime direction, which is the reverse direction. So this gene is located in antisense strand, which is negative strand. How did you know that? Which symbol? Yeah, this one. Yep. And I just wanted to extract as much as information possible by looking at this together. So please just come up with the anything that you see. Thank you for the sharing those kind of tips. And so since this gene is this direction, where is the zoom out? And I would like to explain. So since this gene is a reverse direction, I want to see from right to the left. And then, do you see that the box with a blank, and then the box filled with a color? What's the difference? Pardon? Intron is this line. Yeah. This one, do you see this, my cursor here? Is called 5' UTR, untranslated region, because it's located there. And then start codon should start from this, right? And then there is an intron. And then the second exon is there.
3' UTR, right?
This one should be the 3' UTR. Yep. Now, so you probably know that, okay, what is the protein coding genes, right? But what about this this yellow color? Yep, merged sequences, that's good. What about the blue ones? Yeah. Yeah, it is not going to make a final protein product. So sometimes it's because of a retained intron. Sometimes it's a nonsense VDA decay kind of case. So the blue one denotes it will not get into the final protein product, right? Oh, why? That's actually a good question. Why this nonsense mediated decay or retained intron does not lead to the final protein product? Sometimes stop codon is not properly there, but there are many other reasons. Yes.
So in the dark yellow one, it says merged Ensembl and Hawaii.
Yeah.
So when I was reading about it, it says that it is a common part between two gene models.
Yeah.
So does it mean like if there is any gene which is similar in humans and mouse models, it gets highlighted there? And I also read that they have similar splicing patterns.
Yeah. So the question is about this yellow thing, right? And this is the what is that? The merged data with Havana, right? And I also wanted you to take a look at this one. MANE select transcript. And when you click it, this is the consensus gene model between Ensembl and NIH, NCBI. So the thing that I would like to emphasize here is we can have many different types of transcripts. And some of the things like shown in yellow here is like a merged data set. And the blue one, not the real gene product, protein product, but the red one, the protein product. So they actually not just provide the transcript, but also provide all these the details with that information, and you can actually get all these technical details, even coordinate, sequence, and everything by using this Ensembl. So I don't know what group project you will do for your like a final semester group project, but I want you to at least try to use and get lots of information from here. And this information is changing every time. It's very dynamic. And so, for example, when you actually submit the homework or exam, please provide which version of Ensembl you used. Because if you use some old version, result will be different. So what you got is true under that specification. Yeah, I used the Ensembl 102 version, and then here is the screenshot, something like that. Yeah. Pardon?
How do we go to the old version?
How do you go to the old version? That's a good question. Archive, Ensembl. So there is the Ensembl archive site. Let me show you, share this link. So sometimes new feature is added, and then sometimes some features are like dropped. So hey, try to get the band of chromosome. In some version of Ensembl, it's available, but some version they drop or include. So it's very, very version specific. And so depending on which parameter condition you used, it's not easy to what is that? So when I say that, when I grade this homework or exam, I usually see how you got to the result by reading the snapshots. And because the result can be slightly different unless you use exactly same database or version, right? So please, in the bioinformatics, provide how you got to that result, not really, hey, this is the answer. And if you just say the answer and it's not the correct answer, I would like to give as much credit, partial credit as possible. So provide your reasoning. If your reasoning is correct, I will give the full credit. Okay. All right. Let's take a 10-minute break and then move on to the next topic. All right, are you guys ready? Let's do it together. Yeah, I want to hear your group's opinion. So let's get started with the conclusion or the result. Is chimpanzee more closer to the human or mouse? Which one is closer to human? How many of you think the chimpanzee? Raise your hand. How many of you think the mouse? Right? Everyone think the chimpanzee, right? And we are scientists, and can you provide the rationale? Well, you cannot just explain it. And so I'm very curious of listening the rationale, how you actually can justify your claim to other people. So shall we start with group one? Group one, what's your, how can you justify this chimpanzee is more closer to human? Yeah, feel free to share your screen if you need to.
I pulled up the
Let me stop share. Oh, did I? Yeah.
pulled up the
Yeah.
house, and I decided a lot more overlaps, which you can do here.
Okay.
And this is the human mouse. So there's the overlaps.
Yeah, good job. How about group two? Same or different argument? Same? Different?
Chimpanzees closer than almost the same. For mouse, it's like 4 different.
All right. How about the group three? Do you do the same logic or do you have a different opinion or something different thing?
Interesting.
You guys say no? I think yeah, since you guys share the same, basically, we reached the consensus. Why don't I actually? It's my time to ask the question to you guys. So now you are the defender. So let me share my screen. So you probably did the synteny map, which shows that similar sequences between species. So I think you have done so let's start from the beginning. Ensembl, human, CHR11, go. And you probably come here and then click the synteny, right? And then the synteny between human and mouse is like that. Synteny between human and the chimpanzee is pretty easy to explain. So let's start with that. Okay, which one is the human chromosome and which one is the chimpanzee? The middle one, human chromosome, about 130 or 40 million bases, right? That 135 million. And chromosome 11 is this chimpanzee one. And what is this green color stand for? It shows the matching part of the sequence, similar part of the DNA sequence, right? And pretty much, except for that middle region, it's pretty much the same, right? Okay, so no argument here, but now let's move on to the mouse, human to mouse. Now, let's do the same thing. Can you explain every detail here? So, you said that this one, middle one is the human, right? Why does it have two sides, left side and right side? What is that? Forward and reverse direction of the gene.
No, they have two
Chromosome 19, chromosome 7, chromosome 2, and chromosome 9. Yeah, actually 4. So, did you? One thing we already can see here is the gene or chunk of the part of a chromosome relocation. Something. So, for example, that beginning part of the 5' end, this block, this small block, top one, is similar to that block, the end part of a chromosome 7, right? And then middle part is here. And then the bottom part of around the 20 million position in human, chromosome 11, is close to here. What about that? This one is close to here, but the color is red. Previously, the color was the black, but this one is color is red. What does it mean? It means inversion, the directionality. So to make it similar, you have to reverse it. Right? So that's the red color. And so the genes are the sequence in the forward direction, similar to that, chromosome 7 and chromosome 19, like that way. How about the genes on the reverse direction? Yeah, that's similar to chromosome 9 and chromosome 2. So is everything clear here? What the synteny map is looking for? The reverse direction, this one? Oh, actually, the color is mapped from here. If every mapping from chromosome 7 of a mouse, it has the green. So yeah, that's the origination of the color. And the black color and red color of the lines are for whether you need to map it as is or the reverse it. And then gene has 5' and to a 3' end position, forward direction and backward direction. So that's the like that. So it's pretty complicated, but it actually explains a lot of things. Now you can probably understand better of comparative genomics between human and mouse, which is not that same. And if you do the same thing for C. elegans and some other distant fish, you will see that totally match and not so much overlap. Pardon? Actually the same, actually. If you, if you reverse one of these, it will match. Yeah. Yeah, you have to reverse the, right? Good. So, so by now, I think you can probably explain, understand that chimpanzee, human versus chimpanzee, and human versus mouse, the synteny map. So, you probably get to know what the synteny map is now, and then how to interpret and how to read this. And in Ensembl, it's pretty easy to find the synteny map like this, but you'll see that it's not that easy for like the NIH database. So something is better and easier for Ensembl. Something is better and easier for the NCBI. Okay, let's do one more group project. This time, a gene. Do you know that can be duplicated and located in the multiple places in the genome, right? Or in another case, a gene from human or one species can be found in another species. Synteny map basically suggests that case. So can you try to explain the concept of a paralog and also log with the example of HBB gene to the rodent related species? And I wanted you to be not just a concept, but in the HBB gene, that this part is duplicated to this specific rodent species, rodent-related species, like, for example, like a squirrel, not the mouse, and then in this region. Can you get that kind of detail? So I think now you know that why I didn't share the PowerPoint, right? Because if you just go to the next page, you will see the answer. Basically, try to discuss the concept of paralog and also log, and then one gene of a human HBB gene to the rodent related species actually get particular one. So specific example of that. Yeah, now the time is 7:17. I would like to give 3 minutes. And then, yeah, I encourage you guys either talk or chat. I will just stay outside of the room to expedite, to, like I said, promote your discussion. Do you need more time or are you done? Group 3 is done. Group 2? Done. Group 1? Do you need more time or okay? Are you ready? Okay, let's get started here. Is there any group who want to volunteer to explain concept of a catalog versus ortholog? Yep. Can you actually share that and then share the link to the class by in a chat? Can you explain?
I believe parallel is due to a gene duplication from information gene and or
Is that because of parallel?
yeah,
Orthogonal occur when there are two genes, different genes in two different organisms, but the function is same. And so these genes are orthogonal because mouse and HBB one in humans and HBE1 in human and HBB1 in mouse are two different genes with the same function, but they are also parallels because both of them have an ancestral link, and we got that from the gene tree on Ensembl. Yeah.
Yeah. So this is the order of we found.
Yeah. Okay.
That's good.
Yeah, thank you for sharing this. The synteny that we have seen is that considered to be related to catalog or also not? Also not, because you are looking at different species. So if you are looking at the same using the same species, yeah, it's a catalog. Can it be clear? So the question that I asked was, if I paraphrase again, can you find the ortholog of HBB, human HBB, into into the rodent related? So let's do it together. Ensembl, human HBB gene, and then I'm looking for the gene level. So I click that, and then I'm interested in, oh, sorry. Yeah, why don't I thought I'm sharing. Yeah, sorry for that. And let me repeat the other way how to do it. Yeah. Let's go to the Ensembl again. Human and HBB gene. I'm not interested in the transcript level. I am interested in the gene level. So click the gene. And for this thing, I want to check out also log, because I want to investigate with the in relation to the rodent related species. So it's also log. So I'm going to go, this is a comparative genomics field, and go to the also log. Yep, this is the hemoglobin subunit beta, and then the beta globin, and that's the gene name is HBB. Do you happen to know what is the ENSG stand for? ENS is gene. Yeah. So, and since there is a Ensembl gene, there should be the NCBI gene, right? So there's a comparable one. Oh. Yeah, I wish that this one pop up, but somehow this takes a time. Let me refresh. So, did you actually get the this kind of the also log, right? Everyone get that? And then you can click the rodent and related species, and there are 24 species at this time in Ensembl. And if you check market, and then now you have see the detail of here. I don't know why it shows it twice, but so that's what I wanted to actually show you. Oh, here. If I want to see that only the rodent, there is only one out of 24. 23 are without the ortholog. And the one with the ortholog is squirrel, the Eurasian red squirrel. And the most obvious case is the 1-2-1 ortholog. one gene in the human correspond to one gene in the other species. That's 1-to-1. But in reality, that happens, but on other cases it happens. 1-to-many, and like a many-to-one, and many-to-many. So that's the different types. Do you have any difficulty in understanding 1-to-one versus 1-to-many versus many-to-many? Is it obvious? Yeah. So in that case, ortholog and paralog together. 1-to-many. Many means basically within that same species, it could be considered as a paralog. Okay. Chromosome 11, human, HBV 1-2. This squirrel, many. One-to-many related. If you just combine, if you look at the relation between the species, it's called the ortholog. But if you just forget about this human, if you just look at here, that's parallel. Right? And in some cases, it's possible to many-to-one. Many-to-many. Right? Related sequence, yeah, related. Now let's summarize what we have. This is actually the end of part one reading one. So let me summarize what we covered so far. The key takeaway for the Ensembl. Ensembl provides a lot of biomedical data, right? So, and then, we didn't do that yet. Previously, I asked you to pull out the breakout. Did we do the breakout one case? No. What did we do last week? We pulled out some sequence from the UCSC genome browser, and did we use the some? Which gene was it? BRCA1? Can you do the same thing here in the Ensembl? That should be possible. Yeah. So that is the DNA sequence of a gene. You should be able to pull the DNA sequence of the BRCA1 for human. And if you are done, please raise your hand. I would like to check your speed, like 100 meters, okay? Okay. Done? So how many of you finished so far? Please raise your hand. All right. Why don't I actually do this together with you so that for those who might have a difficulty can do the same thing with me. Again, the same thing. Ensembl, human, BRCA1. human gene, that's fine, right? Yeah, where should I click? Yeah, where is oh, here, yeah. Yeah, here is the left side, there is a sequence menu. And now you will see that here. So this is a FASTA format, starting with the greater than sign. The pink is the all exons in this region, and then the red one is exons of BRCA1, because multiple genes can be in that same region. So and then BRCA1, is that the gene located in the forward direction or reverse direction? It's reverse direction. So let me go to the bottom. So this is the exon 1. And then the exon 2, like that. And then these are the intronic sequences. So this one, even though we didn't ask, they highlighted what is that, the exons. So if you in UCSC, you have to check mark it. Anyway, it's about the same information, but something if you prefer this way, yeah, you may want to use the Ensembl. But if you prefer, I would like to customize myself, then you might want to use the UCSC. Okay, let me come back to the take-home message of key takeaways for the Ensembl. So now we just retrieve the DNA sequence of a gene, which was pretty easy. Just type the gene name and browse the left menu, and then click the sequence. That's it. And then, do you remember we found the 6 isoforms transcripts of HBV gene? And we even got to know which one is the protein coding, the red one, and which one is the pseudogene, the blue one, which is not actually leading to the protein. Yeah. Two of them are protein coding, the red one. Yeah, but this one is the merged data. So you have to be careful. You have to take a look at it. Yeah, but this one seems like a protein coding, different from this. But you have to double check. No. Based on this, we have 6 types of mRNA. But if you check out how many eventually code for the protein, then the 2 of confirmed one with the 2 of merged one. So total 4 possibilities. And 2 are like but you have to be careful on this merged data. So that's why I'm saying it. You have to take a look at it.
It's the same in the market.
Havana. Yeah, Havana, the other database. Yeah. So yeah, so my point is, for when you look at the merge data, be careful and check out the source. Yellow color. Yeah. 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 This one is mostly due to the some like a termination codon problem or like that. And how many coding potentially 2 to 4 proteins. Yeah. Yeah, you have to double check. Yeah. And then this data is always changing. In the next release, I don't know what will happen. So that's why I'm saying it. This is true with this parameter and this condition and this version. And how actually you can check in your own biology doing RNA-seq. Yep. And even though there are 6 types of the mRNA, not all the mRNA may be expressed. So it also depends on the condition of the tissue specific. So far we got to know that the evidence for up to 6 different types of RNAs. But that doesn't mean that in your tissue you find all of them. No, it's not. Yeah. Right. Yep. So in Ensembl, you can find a lot of other information easily. That's why we are using this database. Can I just move on without some break? Even though it's we are reaching the 7:50. Yeah. Okay. Now the Biomart. How many of you have experience of using Biomart? Please raise your hand. No one? That means you don't know the Biomart, right? Yeah, that's fine. Okay. All right. That's fine. So, one of the good things. Did you find the here? And if I just say that, yeah, you can easily check the synteny map, also log open chromatin region, and then the isoforms and the sequence in the Ensembl. You probably found that, oh, wow, can I do that? But by now, I think you can do it, right? So even though you didn't use a Biomart, it's not that hard. But I would like to explain what the Biomart is first. Then let's try to use it. So far, you click somewhere in the Ensembl database and retrieve the data, right? So if you want to get the DNA sequence of a gene, click here, type something, and then click here, and then you will get it. That's way. So the way how you retrieve is based on clicking so far. Do you agree with that? That's not bad, right? When you try to get the information one by one. But if you try to get some thing in a batch, meaning that a lot of things, related information, or file or something, clicking sometimes is really annoying. Yeah. Is there any, how many of you know the query? No, you don't know? Okay. How many of you know the MySQL? Okay. So is there anyone who can explain the MySQL?
So database management software.
Yeah.
It's a coding language basically, which gives commands to edit, modify, and delete, and retrieve the
Yeah.
database with a structured query language. SQL stands for structured query language, and it is a way to manage structured database.
Yeah. Ensembl is a database. Yeah, basically. One way to access the data is by clicking, but since it's a database, you can use some language to retrieve the information. Query-based, right? So Ensembl allows you to retrieve basically the way kind of what do you say? That kind of a query language-based way. So you can actually have some like a query table, and you can click to check mark it, or you can type it, and then query is passed to the MySQL database, and then that retrieves the data. Instead of click top left corner, and then click the right side of a thing that's written like that. Instead of that approach, the hyperlinking, you can retrieve the information by the query style. Did I explain? I hope that you understand what I try to explain it. Basically, I'm trying to say that, tell you the difference between click the top left corner, then click the bottom right side. That is one approach that you just have done. But another one is the using the query language to retrieve. It's basically the same information. So that's the Biomart. Yep. But yeah, why don't we actually do the group project? Ensembl is assembly of many databases, small databases. So far, we want to use the Ensembl gene. You know, HBB gene is listed in the gene database of Ensembl, right? So can you query against Ensembl genes? Ensembl is a gathering of all these, and you want to particularly query not the transcript database, but to the gene database. And for example, HBB gene in the gene database, like that, right? So you want to query into the gene database, and version is important. Let's try to use 110, which is the current version as of today. And for 1 month later, they will probably release the newer version, 111. And 4 months later, every 4 months, they release a new version. Yeah, so, okay. And I would like to query against mouse. And it's very important which version. Genome Research Consortium's mouse, musculus mouse, version 39. Let's use that. Which reference sequence that we want to use, right? And then mouse should have a chromosome 11. So we want to confine the chromosome 11. Do you remember we previously done in the search box, we typed the CHI11, but instead of click that search box and typing it, let's use the query language. So like that. And then HBB, how many transcripts did you find that the HBB has for human? 6. But in chromosome 11, there are some genes that might have more than 7 transcripts. So HBB is not counted for that, for the human. But there should be some genes, right? they may have more than 7 transcripts. And then I also want to limit the genes, the reference sequence database is an NIH's database, NCBI database. I want to have genes that's also curated in the NIH's RefSeq protein database. So I want to make it sure that this gene is not just in the Ensembl, but also in NCBI protein database. And I want to see all the basic features, attributes. With since I checked this RefSeq protein sequence, I also want to get the output of the RefSeq protein ID. Can you do that by clicking the manual way that you learned so far? I don't know how to do that, but if you happen to know, please let me know. I can learn, but I don't know how to do this kind of complicated one. But if you use a query style, you can actually do it. And if you use a Biomart, which is a interface of the Ensembl database, you can use a query style, and then you can execute this kind of a complicated condition and retrieve the right information. So what I want gene name and transcript ID, and in addition to, I want to also get the RefSeq reference sequence database ID in the NCBI, NIH. For genes in the mouse, chromosome 11 with a more than 7 transcript count. And this gene should be cross-compatible in NCBI, like a protein database too. Do you happen to understand what I'm trying to find it? So basically, in short, in a one sentence summary, I want to get the very complicated information, which is not easily accessible by using the way that you have done. But by using the query style, you can actually do this. So I will give the about 5 minutes, and then we'll come back, and then I will just stay outside so that I'm not going to do any other things. But yeah, one thing I found was if I'm here, you guys are more quiet. So yeah, why don't you have some free time, and then feel free to discuss, and I will come back. And if you need more time, I'll give them more time. And I understand that this is very easy if you know how to do it. If it's not easy if you never have done it. Yep. I will come back in 5 minutes. So how many groups get the answer, like more than like the what is it, the this one, group 3? How about group 1 and 2? Did you get that? All right, let's go together then. So basically we want to get the candidate. I want to get the some gene name and transcript IDs with a protein ID that has more than 7 transcripts in chromosome 11 for mouse. But let's do it. So let's start with the Ensembl again. There is a Biomart. Do you see that? And making sure that it's a mouse, they click to choose the data set. Yeah, latest one, Ensembl genes 110 version. And then it's making sure that this is a mouse, right? And I want to restrain myself in the chromosome 11, which is called the filtering. So I want to click the filter and chromosome, go to the 11, right? I check the chromosome 11, all right? And my main question is the gene that has more than 7 transcripts. So do you see that the transcript count column? Highlight that and more than 7. So if you highlight it, deselect it, this is gone, right? But when you select it, now it's there. If you want to have more than 5, if you want to have more than 7, yeah, you can have that, right? So far I specify the 2 conditions. Now I want to specify the last condition, which is in NCBI has a RefSeq peptide ID. So in NIH's database, they should have some relevant peptide ID. So that should be here. If you look at the gene section, there is a limit to gene and with a RefSeq ID, peptide ID only. So by selecting that, it comes up here, right? And so now I'm done with the filtering. And now I would like to specify the output, which is the attribute. And I want to have a basic, the default, and one more thing additional, which is external reference. I want to have a NCBI's RefSeq peptide ID also shown as an output. That goes to the attribute section. So filter. So basically, it has a 3 data component. Data set, which database are we using? Filters, meaning that narrow down the condition, and then attribute is the output section. Is it clear? Pardon? Attributes are what kind of output you want to get. Do you want to get the transcript ID? Do you want to get the gene name? Do you want to get the Ensembl gene ID? Do you want to get the NCBI gene name or like that? So that's the attribute. Once you are done, click the count to make sure how many candidates you have. Yes, 331. And click to get the check mark. And then you have many like solutions. And if you want to have save it as a tab separate value file, yeah, and you can click go and you will download that as a file. If you want to prefer the comma separate value file, CSV, you can click and go. But did you notice that a lot of redundant things are shown? So even though 331, they are not all the independent ones. So if you want to simplify to get the take out the redundant one and just leave the only one. So if you have a 10 same thing, take out 9 of them and just leave 1 unique one. Then you want to check that when you download it, and then just click to go and get that marked export.txt file. So unique result can be checked out here, or after download, you can use Excel to do that. It doesn't matter. So if I summarize it, in the beginning, if I just tell you, hey, get the gene names that has more than 7 transcripts in mouse, chromosome 11, that might sound very difficult, but by using the query language built in, you can easily do that. Any question here? So it's not that bad, right? Yeah. Can I move on to the Biomart? Right. Pardon? Unique is removing the duplicate only. Just leave the only one unique one. And which will simplify a lot of your work because you don't want to have 10 or 20 the same record. Right. Now, so far we talked about the Biomart, which is the query language-based data retrieval from the Ensembl. Now let's talk about the Biomart. Did you notice the difference between previous Biomart and this Biomart? Do you see this red color? Not the the R. Yeah, this is really funny, but if you type this way, that is you are using this web interface to do the query. But is it possible to use R for doing the same thing, which is a Biomart? But in that case, you have to write like this way. Okay, and this is R-based. From R package, you access the Biomart, and Biomart is located in the Ensembl.org. So that is a flow. So this R part is the new thing, or that's the differentiating thing. Why does it matter? Because you already know how to do this Biomart in this thing. But why should I learn the another way of doing it using R? If you have a large database, yeah, if you just do only one task, yeah, I probably use this regular Biomart, the web-based. But if I do 1,000, like slightly related, I would probably want to code. And in that case, if you can do it using R, you can easily code and you can automatize. So there is actually advantage of learning how to retrieve the data from Biomart using R language, right? So before we jump in, okay. Let's get started together. And then anyway, we need to install this software, Biomart. It takes about 3 to 5 minutes. In that time, we will come back here, and I'm going to address how to do that in the web-based. Let's do it together too. So shall we directly jump in the Biomart in R? How many of you installed the R locally into your laptop? Right? So you have an option. You can either use your own version, but I recommend that you to use the R Cloud because everybody is on the same setting, and it's easier for me to troubleshoot or manage. When you use the your own R in your own computer, I don't know all the settings that you have done for your own computer, so troubleshooting is somewhat tricky. But if we use the R Cloud, everybody is basically on the same page. So for the class purpose, I would like to use the R in the cloud. But it doesn't matter. The result should be the same whether you use web version or your local version, right? Now, can you go to the R Studio Cloud? This is the site, and let me copy and paste this link. Why don't we do that together? Can you Google R Studio Cloud? When you Google the R Studio Cloud, it should give you Posit Cloud. They changed the name from R Studio Cloud to the Posit. Did you get this link? Can you go to the site? Do you come to this kind of site? Can I assume that you have at least one Gmail? Can you click login? You don't need to sign up. you can authenticate by your Gmail. So please log in. Can you use the login with Google? And please use your own like a login, the Gmail to log in. And when you have it, can you click the new project? When you get here, can you click the new project? So, and then you want to create a new project of new RStudio project. You are not doing the Jupyter, you're not doing the Git, but you want to use the RStudio project. Let's survey. I would like to know your proficiency for R. I'm virtually the first time. Please raise your hand for the R. Right. Okay, and how to know your familiarity of the R? Okay. Do you know what the C function is in R? Please raise your hand. Okay. How many of you actually know the making the list in R? Okay. How many of you have experience of installing the R package? Okay. And how many of you have experience of using Biomart? Probably no one. Okay. And how many of you have experience of using ggplot? Okay. And how many of you have experience of using the t-test in R? Right. Now, basically, I get to know. All right, thank you. Okay. Oh, how many of you have experience using R script? How about the R notebook? Okay, good. R notebook? Markdown.
Ah, yeah.
Yeah, Markdown language. Right? Okay. Yeah, based on your like thing, okay. Everybody has this page, right? This is the R console. You can type the R command directly. But if you want to have a record of it, what command you typed, you'd better have some records. So can you click this icon? This icon. Now you are looking at the 3-window system, the R console in the middle, and then 2 other windows. But I prefer you to make a 4-window system by clicking this icon. When you click that, do you have a 4-window system, right? Here, this is a basically notepad. You can type it, and then you have the record of it. Okay, and so if you do the 1+1, and then highlight, and then run, then it is going to be pasted into the R console, and the answer is 2. Let me just do one more example. 2 to the 10. Highlight it and click run. You get the 1024. Everybody on the same page? Okay. Now let's do the real thing. Now you came to this Pajit Cloud and log in with your Gmail and started a new project, right? It's time to install Biomart. the first you want to install the bioconductor manager. Biomart is hosted in the bioconductor repository. So you need this kind of a helper. So let me paste it into this is a one-line command. Can you run this command to install bioconductor manager, BioC manager? This is one line code. So go here, and then you just highlight it and run it. If BioC manager, bioconductor manager is the exclamation means not installed, not there. That is not. Then execute the following command: install the packages, particularly the bioconductor manager. That's what the syntax is. So did you run that? And did it run something and then now you have a new prompt? That means it's installed. So it's why don't I give us some let's install bioconductor manager. Bioconductor is a repository name where it hosts the many packages like this Biomart. Install bioconductor manager software. Step 2, use this BioC manager to install Biomart R package. The command is this. Highlight it and then run it. So do you understand these two steps? Before you install the Biomart software R package, you want to make sure that the bioconductor manager is properly installed. Then using that BioC manager and access the particularly the install the function in that BioC manager, then you install that Biomart R package. That's what I meant by do you have experience of installing the R package in R using the command line? Using the command, not really the token. So this will take about 5 minutes. So I would like to do the following thing together while we are waiting. So after installing the and then installing this and being able to use this, what we want to do is basically we want to extract a gene name and transcript ID in Ensembl that correspond to NCBI's gene ID 6928. So you know the NCBI. Let me explain the step by step. You know the NCBI NIH database, not the Ensembl, NIH database. There should be some gene whose ID is 6928. There should be some gene, right? What is the gene name of that in Ensembl database? What is the transcript ID of Ensembl? So that's the what can you actually think about how to actually retrieve that in the Ensembl? Can you retrieve by clicking? Maybe trial and error. But if you use that query thing, Biomart, you can easily do that. So shall we try to do that together while we are waiting for installation? So let's go to this thing. I will give the about 5 minutes. Can you run the Biomart on the human database? And then, so let me explain. So you know this interface, right? Biomart interface, web interface. The goal is try to find the gene name and transcript ID that correspond to the NCBI's gene ID 6928. Can you use this kind of Biomart to do that? So specify human gene database, HG38, and then the way how to filter is NCBI gene, and then ID is specified as 6928 there, and then the attribute that I want to see is the transcript stable ID and HGNC symbol. HGNC is the common gene name. So this HGNC symbol will tell you the gene name, and transcript ID will tell the transcript ID. How many results did you get? Is there anyone who got the result? Did you get this kind of result? Different? Is there anyone who got this result? Last two, attribute. Oh, you are looking at the top 10, right? Can you change to see the more than 10? Can you show the top 20 result? It's a 12 result. So did you get it? Who else get this? You get it? Yeah, I will go over anyway from the beginning, so you will actually get that. So yeah, yeah, don't forget the what we are trying to do. There should be, I want to find out the gene name and transcript ID for NCBI gene ID 6928. That's what I want to get. So shall we try to do this together? So let's do the same thing. Ensembl. And I want to click the Biomart. I want to click the new to refresh. Right. The database is the latest Ensembl genes. The particular data set that I'm looking for is human genes. GRC stands for Genome Research Consortium. H stands for human. Version 38. P14 means minor update, 14th version. Right? Now I got the data set, and let's apply the filters. The filter, I want to have the NCBI gene, formerly untraced gene ID. That should be corresponding to the 6928. So I have to find it where it is. Is it in the gene? NCBI gene, formerly untraced gene ID. Input external reference ID. Here, NCBI gene, formerly untraced gene ID. Let me repeat. For the filters, I go to the gene, and I want to provide the input from the external reference ID, which is the NCBI. So click that and choose the NCBI gene, formerly untraced gene ID. And then I type the ID name, 6928. Did you find this? Okay. Now I'm done with the thing, and now I want to specify the output, which is attribute. So output, I want to have a transcript stable ID and HGNC symbol. I want to deselect everything except for the transcript stable ID and the external source HGNC symbol from the external sources. And when you're ready, click the result. and only top 10 shows, but I want to see more than 10, then show up to 20. Then now I have one gene, HNF1B, with this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this gene has more than 7 transcripts, which is located in the chromosome 11 of human. Yeah, question? Oh, these two? Yeah, I click the account, and then it actually turns out to, what is that? The 2 count of among the 70116 genes. But actually, if you look at the result, it corresponds to the one HGNC symbol with a 12 different the transcript ID. But let me show the this just one moment. So what I did was, it means that there are 2 genes out of 70,116 total genes available in the Ensembl database. Why it's 2 genes, not 1? When I put the gene stable ID as an output, if you look at the result, even though common gene name is everything is HNF1B, gene ID we have 2. One is the ending with a 194. The other one is ending with a 410. So the top half of the Ensembl transcripts belong to this particular gene ID in Ensembl, and the second half belong to the another gene ID in Ensembl, even though the common gene name is the same. 2 different Ensembl ID exist out of 770,116 gene IDs in the Ensembl. 2 different Ensembl gene ID, but 1 common gene name. Is it 1 gene or 2 genes? No, I'm asking. HGNC symbol is the common gene name symbol in another database, outside database. So this is the nomenclature database of this Hugo gene database. So according to this database, it's 1 gene. But according to the Ensembl, it's they have 2 separate IDs. Yeah, so databases are not exactly matching each other. Yeah. Yeah. Yeah, but we are actually looking at the Ensembl gene. That's why we have 2. Yeah. Why? Yeah, since this is the Ensembl, yeah, this is should be based on Ensembl. Yep. Oh, you actually specify here. attribute section. User, yeah, that's right. But this number is based on Ensembl database. Any question? So, how many of you eventually get this? Right. So now you know if you want to retrieve, hey, genes that has more than 7 transcripts, or try to find out what is that? The gene name and common gene name and transcript ID that has corresponding to the NCBI specific NCBI gene ID. You can retrieve it, right? Is it possible to do this using the command in R? That is the next topic. So everybody, how many of you actually completed up to here, installing this Biomart? Everybody done the Biomart installation? Did it work or generate error? Everything works? Good. Let's move on to the next step. Load the Biomart into the RAM by using the library command. Library, open parenthesis, Biomart. And then why don't I give the command in the chat? So the next step is load the Biomart R package. The command is library Biomart. I just put that into the chat. So let me highlight it and then run it. Did you run that? So if you run it without any error, that means this is ready to be used, right? Let's do the line by line. I'm going to type the following command. Step number 4, use the Biomart to get the output in the following 3 steps. (4-1). Let's specify to use Ensembl and call it Ensembl. Ensembl equal usemart of Ensembl. So is it obvious or do you need a more explanation? Why don't I actually give the this again in the chat? Yep. So let me highlight it and then run it. So now we by calling Ensembl the variable, we are going to say it. Yeah, I'm going to use the Ensembl database. Then step 4-2. let's use Homo sapiens gene ensemble from first one is actually optional. You don't have to use it. But second one, from the Ensembl BART, use the this Homo sapiens gene ensemble particular data set and call it the ensemble. Oh. Mart, ensemble. Let me just copy again, sorry. Yeah, I just put the new one. I typo. If I the ensemble, this should correspond to that ensemble. So I drop finally use the get BM function, get the result from Biomart function to get to get transcript ID, which is and common gene name. And let me paste it to the chat. So the actual command is, let me paste it again, actual command only. We want to use the get BM, get from the Biomart function. We want to get those attributes. The output variable is the ensemble transcript ID and HG and C symbol. But we want to filter it based on untranscript gene ID, particularly the value 6928 from Ensembl Mart. So this is the core command, the most important command. And when you do that, you probably get the this 12 output. And if you compare with the previous web version, the result should be the same. So if I explain this again, the syntax again, we specify to use the Ensembl database and then call that as an Ensembl with the E. You can type any, this is just a variable to denote this is Ensembl database. And this variable goes into that, the Mart. Mart equal Ensembl means, yeah, we want to use the Mart Ensembl. So it's a shorthand. And we specify the data set, Homo sapiens gene Ensembl data set in the Ensembl database. So do you know the difference between data set and the whole database? Whole database is Ensembl, but specific data set that we are accessing is Homo sapiens gene Ensembl data set, small subset. And then finally, we want to use the get BM, getting from the the Biomart, which is a R function. We specify attribute and then filters. Attribute is basically the output. Which output do I want to get? Ensembl transcript ID output, and then HGNC symbol, which is a common gene name. And then the filtering condition is based on untrace gene ID. Value is 6928, marked from the Ensembl that we defined here in the step 2. You have a question? It can be switched, no problem. So did you get the same result as before? How many of you get the result? Please raise your hand. Okay, most of you, good. So what do you think? Using the, so let's see a kind of a backup. So you just use this command to get this kind of R output, which is the same as the web version. So when you use a query-based like a Biomart, you can either use web version or you can use the R version, which is the same. It doesn't matter. But if you want to code, you prefer to use R version. But it's just one or two, you can use the web version. From my point of view, I don't there is no big difference. Whatever it's easier, just use that. So take-home message is for this part. If you want to retrieve the data systematically, you may want to use the some query-based method. You can use a web version of a Biomart or R package version of a Biomart. Right? And this is basically the end of the content for this Unit 4 today. And before I move on to the group project, do you have any questions? No? Pardon? Oh, the group project. Let's go to that together. Yeah, PDF is correct. Oh, really? Oh. Let me check that. Thank you. Just give me 1 minute. Let me check that. You mean the group project one, right? Yeah. Yeah, I tried to refresh to the 18th. Can you double check? Does it update it automatically or still due date something problem? It didn't change? It changed now? Okay, good. Yeah, sorry for that. The date was not like properly configured. So do you have any question about the brief summary of what you have done? Just summarizing the one paper together. The purpose is you want to use this as a benchmark paper and you want to do the group project better than that. Now let's move on to second the thing. Let me share my screen. So let's go to the canvas, modules. This is 18th. No. Yeah, let me double check the date. Yeah, thank you for saying this. So date should be 25th, or what is it, right? 25th. So I updated. Right. This is the second step out of 10th step. So this due date is 25th, which is all right. Since this is not really immediate, so you have many days. I just want to give us some lead so that you know what's going on. So today is the 14th. By 18th, you just summarize the benchmark paper, which is just the summary. And again, just don't use the ChatGPT to summarize. As long as you don't use that ChatGPT to summarize, that's fine. But when you summarize the paper, be aware that the paper is usually how many of you have? Did I ask this question right? How many of you have experience of publishing a paper? Right? Probably one or two people have that, and then was it the first author Yes. or was it first author? Second, okay, right, okay. Only two people have the experience. So I think I would want to say this: the scientific paper published in the journal has a very specific format. Every scientific paper is almost the same. Title, author's name with affiliation, abstract, introduction, method, result, discussion, and optionally conclusion and references. That's it. Basically, that's the kind of universal format. And the most important thing is in the paper is main hypothesis. So when you actually see the paper, you have to be very careful to read what is the main hypothesis of the paper. So the paper that you chose should have at least one hypothesis to prove or dispute. Most of the case prove. But in our class exercise, group project, negative result is fine too. You know what I mean by negative result? Meaning that you may dispute your hypothesis. Like saying that, for example, we hypothesize for this novel SNP play important regulation role for Alzheimer's because of Wnt signaling pathway involved, and then that make us some hypothesis. And you perform the analysis based on the publicly available data, and you either prove or dispute. So when you actually read the benchmark paper, making sure that you actually get all this. What is the main hypothesis? How the author tried to prove it? And then is there any like problem in logic or any wrong the steps? So you can do the rebuttal or you can fight back to the author. And if your group is doing the same kind of work, how can you avoid those kind of pitfalls or like that? So I want you guys not just simply read it, but critically read it so that you can excel even that publicly published paper reviewed by the experts in the field. Right? So that's what I actually want to see in the summary, not just like a regular kind of simple summary, but your critical summary. That's the kind of what I want to see. Okay. And given that the next assignment in this unit is it's time to choose the novel SNP. So now it comes to the biology here. This is very important thing. Usually in the class group project, professor usually say, hey, study on this. Give the material and you basically combine yourself. But not this. You guys decide your topic. If you are interested in Alzheimer's disease, study for Alzheimer's disease. If you are interested in a cancer, like a tongue cancer, yeah, study for that. If you are interested in any like a genetic related disease, choose that thing first, the clinically relevant thing. But did you see that early 3 billion SNP in the course of development for humans? So out of 3 billion, you will probably have about 1 billion. And which SNP do I have to choose? That is this. So your group discuss it online or in person, doesn't matter. And which subject do you want to study? Try to identify the benchmarking paper, not just one, but the one is kind of shows the typical procedure. But when you do this, I want you to find out if I'm interested in the Alzheimer's, identify which gene is relevant to the Alzheimer's. That is APOE4, for example, or something. And then check out the many researchers. Eventually, you want to cite minimum 30. Okay. That means you better read more than 50 or 60 papers in this semester. So benchmarking is just to show you one representative flow of this paper. But when you reach that stage, you have to read many papers. But you guys decide which phenotype you want to study. And the SNP that you chose for the example of Alzheimer's, APOE4 is one of the genes known to be closely related to Alzheimer's. And you have to know how many SNPs in the APOE4. You guys talk to each other and then choose the one that is not extensively studied. There is no absolute the criteria. You guys decide. If you happen to choose the SNP that's extensively studied, it's not easy to make a hypothesis, novel hypothesis. That's why you want to choose some. But one tip is if possible, try to exclude intronic as much as possible, because not so many things can be bad unless it's relevant to the non-coding RNA or long non-coding RNA, or it's in the super enhancer region and those kind of particular case. But if that is in those regions, yeah, you can choose it. So when you actually choose that particular SNP that you guys want to study, think about the hypothesis too. You don't have to complete the hypothesis by this week, but thinking about the main hypothesis actually helps you to choose the right thing. Okay. But I don't. This is not what I want for example. BRCA1, and let me just choose randomly. No, don't do that. But if you do that, it's really hard to make a hypothesis. So read a lot of paper about the BRCA1, and then choose carefully the SNP that's not extensively studied, but it is still in the exonic region and change the amino acid or something like that, so that you can make a good hypothesis. Does it make sense? So, yeah, so you have some time now. So you can think about it. Okay. Do you have any questions? No? Yeah, again, when you submit, you just submit only one master PDF with all the screenshots there. And then if you guys want to grade it individually, you can, what is that? Use different colored fonts. So green color for person one, red color for person two. Then I will grade individually. But because it's not easy to make an equal contribution. Some person contribute more. That's inevitable. That's why you have a first author. But if your group really wants graded the same way, believing that everyone's contributed equally, that's fine too. In that case, use a black font. Then I will understand. Okay, you guys wanna grade it as with the same grade. But in that case, you shouldn't complain about I worked more, but why my grade is the same, because that's what you choose. Is it clear for the grading too? Okay. Now let me stop recording. And then